quick studies.
Thank you. So I'm going to present a talk which has a little formula, probably not much. It's mainly about some concept, and I have a bit biology to share with you. Okay, I got time to revise my transparency a little bit after sitting here since yesterday. There have been a lot of talk, and I gather what particularly in this place, we know that calculus is a subject about change, and life science is certainly about change. We do not change, there's no life. And my entire talk is in fact about various aspects of the change. All right, so this is a slide I add here. We hear some comment about what the size inverse regression was doing. So the forward regression was teaching us how the output changes in response to the changes in the input. And so you have this forward paradigm. And when the dimension is large, you may try to reduce the dimension of the x. And one typical way of doing this is doing the principal component analysis and so on. But there's a danger of the loss of information. And so a reversal of that paradigm is to start from the y. So instead of asking how y changes in response to the changes in the x, we are now asking how x changes as y changes. So this is the sort of the intuition behind the size inverse regression. And there's a theorem connecting the forward and the backwards regression. Okay, so we know that regression is about, it's a theory about describing the change. And this gentleman, Francis Galton, who introduced the regression analysis. And actually, I would like to point out, in case of people forgot, he is a half cousin of Charles Darwin. And so, in fact, his examples he's tried to explain is about the genetics, how the size of the seeds evolved through successive generation. And he established that the regression slope equals to the correlation if you properly standardize it. So this is again just iterating that the correlation is a built-in block of many things in multivariate analysis, in cluster, classification, and dimension reductions. And the things I'm going to talk about, liquid association, is in fact about the change of correlation pattern inside the scatter diagram of a pair of variables. So I'll make that clear. And I give 2 slides to summarize what I do. And so if you can understand of this, you probably don't have to sit for the whole hour. So basically, that in the typical micro data analysis, that people doing a lot of clustering analysis. So basically, it's look at the paired genes and quantify its correlation and use that to group the genes. And we have a way of doing things differently, and it's particularly that we think that quite often that the converse of this statement is not quite correct. That is, usually, although correlation is an effective way to spot functionally associated genes, but in fact, the most functionally associated genes do not have a correlation, and there are many reasons behind it. And one is the multiple roles of most genes can play, and their roles can change. There's some hidden variables or hidden states changes. And liquid associations, as depicted in this little diagram here, is try to capture the hidden changes. Okay, so it involves another variable, and I will try to make a good story about that, how important that is. And since the liquid association involves three variables, so there will be a question of computing. How are you going to compute the liquid association score for the n-cubed triplet? And just to give you the numbers, n equal to 6,000, that's in east, you have about 36 millions. And if you have 50,000, say arithmetic probes, then you're talking about 20 billion trillions. And so the time is really pressing. Okay, so the rest of this, you probably wouldn't understand it right now, but we'll try to get to those as much as possible. And it has a lot of applications involving, say, finding the disease candidate genes. And the technique can not only apply to the microarray, but you can have other type of the gene expression, genomics data such as array CGH and so on. So I might not have the time to do it. Okay, so we are talking about the gene expression data as a primary example here. So typically, you have many genes measured at different conditions. And as I said before, the clustering is the key in making these techniques so popular among the biologists, because after you suitable group in the genes, and then biologists can pop into its functional in each clustering, and they say, hey, for this group, the genes, their functions, suppose they are known, they seems to be making a lot of sense because they're essentially doing the same thing. So this is the biologists way think that clustering is making sense. This is based on genes with a high degree of expression similarities. They may form structural complex, or they may participate in common pathways, or in terms of gene regulations, they probably share some common upstream regulatory elements in the. Yep.
Can you define what you mean by profile similarity?
All right.
Give some idea quickly, just so I can be educated.
No, I'm not trying to educate you. But the expression similarity is, in fact, a term that without proper definitions, it's just the biologist will say, hey, this profile looks like that profile. That's the same thing. So it means that if one gene is highly expressed, the other is also highly expressed. Now, when that genes under certain conditions slowly expressed, the other genes are.
So it's a subjective impression based on the data,
right?
Not something that comes from outside the data.
Yeah.
Okay. Thanks very much. That's what I need to know.
Okay. Yeah. Well, in this world, that I think the biologist and the statistician or mathematician are trying to educate each other. And it always starts with some sort of a common language, and you try to formulate it in terms of the right terms. So there are certainly many measures of the degree of the similarities, like you were talking about. You can use a generalized correlation if that's you can persuade the biologist to accept that. But typically, you just use the Pearson's correlation. And this is a nice picture, just show you how different parts of how different protein putting together as a protein complex to produce a sort of an enzyme complex to carry out an important function. In this case, it's called the mitochondrial ATP synthesis. And the amazing thing is that there's a high degree of similarities across the species. This one is for the E. coli, and I think this one is for some monkey or you know, mammalian. So this is just a little exercise. When I teach, I just try it out on students. And so you give you 7 genes, and you can pop out, say, their gene names. It's called the MCM1, MCM2, 3, 4, 5, 6, 7. And assume that you don't know what the heck of them are, and you simply take a set of the gene expression data and look at their profile. And this is just the scatter plot. So each dot represents the expression of 2 genes. So for example, this one is this genes against this genes, and each dot is measured at certain conditions. So the scatter plot, and you can see that all this, if you look at this part here, it's all positively correlated. So indicating that MC, MC, M6 and MC7, they're probably functionally associated, and this one is also. In fact, the rest of them are quite similar until you hit this guy here. This is another MCM1 here. Then you see that all of a sudden this nice correlation is gone. This part is symmetric. So you can infer that the function of MCM1 probably is different from the rest of those MCMs, although they seems to be named in the same family, but in fact they are not. And so the truth is that the MC227, they form a hexameric complex that binds the chromatins, and it's when you start with replicating the DNA, it has to start from somewhere, and this complex just binds that to initiate the replication. So its function is in the replication. Now the other one is quite different. The other one is a transcription factor. So it activates the gene expression. It does not involve in duplicating the chromosome. Okay, so if you just take, this is the problem with the biology. When they see some successful examples, they try to blow it up and say every case is like that, until people find a counterexample. And in fact, that well, this is actually not our finding, but it's been known by many others also. That is, the expression profile of the majority of the functionally associated genes are indeed uncorrelated. And so the sad thing is that biologists simply report the good news and forget about the bad ones. The microarray data are too noisy, and usually you can bring that, but in fact, that should not be the only reason. The two reasons is that the complex biology involved. So here are a couple of reasons that proteins rarely works alone, and they have multiple functions. And among the cell, inside the cell, there are many biological functions going on simultaneously, and those have to be synchronized. And And there's also other things like competing resources, and also we are measuring the gene at the expression level, but in fact, as the previous speaker pointed out, it's the protein that carry out the work. So how does you go from the expression to the protein? That's another story. And also protein has to undergo in some modifications and so on. Now, what I would like to address is, well, I forgot to tell you the last part here. We know that there are certain protein whose function is to regulate the gene expression. Those are called the transcription factors. But usually, I think the people know that some transcription factors are activating the gene expression, while others are replacing it. So we have the activator and the replacers. That's probably most people know. However, they forget that transcription factor, just one transcription factor, sometimes can act both as the activator and replacers, depending on the situation. So if you think that a transcription factor is an activator, and you think it's a protein level expression level of the X, and Y is the target genes, then if it's an activator, then you would expect a positive correlation. If it's a replacer, then you will have the correlation will be negative. Now, what will happen if it is both activator and the replacer? Well, depending on how you set up the experiment. If your experiment are not properly set it up, it makes up two different conditions. You probably don't see the correlation. And what I said here is not just the statistical speculation, but it's real. There's a gene, there's a transcription factor called the serum receptors, which actually serum hormone receptor, which act like that. And the biology is a little bit deeper, but I think it's conceptually it's not that hard to explain. So I'll try it here. Well, in the way, well, this is the work in the eukaryote. I think in the prokaryote is probably simple. So why the people always have the transcription factor dichotomized as the activator and the replacer, because in prokaryote it's much simpler. But in eukaryote, the DNA is wrapping around a protein called the histone. And histone, because it's blocking the way, so its role, well, there are many roles, but one role is to inhibit transcription. Now, if you would like to activate the transcriptions, you have to get rid of this the histone. And now, unfortunately, the histone has a lysine chain which is quite sticky, and those are why this is wrapping around. And so, in order to release the lysine chain has to be insulated, and which is by adding an acetyl group. And so that process is called acetylation. So there are enzymes doing that. Without the serum hormone, the enzyme, which is called the deacetylase, this type of the enzyme is doing just the opposite. Its function is get rid of the acetyl group. So the histone getting very sticky. So it's very tight here. So here is a normal situation. You still see some loosening here, but it now becomes very tight. So all the transcription totally stopped. And if the hormone comes in, then there will be a change of the conformation on this part here, and would be this deacetylase will be kicked out and replaced they will recruit another type of enzyme called histone acetylation. Histone acetylate, and it will recruit the acetyl group and attach to the lysine chain, and all of a sudden you see this wiggling around. That's the DNA got opened up, and that's how the transcription can carry on. So what we're seeing is that depending on the level of this thyroid hormone, a single transcription factor can act either as an activator or replacer. So if we are talking about the gene regulations, it's very hard to see whether you should see a positive or negative sign in there, and that's the whole complexity. And what I explained above is just one of the difficulty issues. But in fact, as you may already know, that what we have is the messenger RNA data. But on the behind it, there are so many other stuff that we cannot measure. So there are important issues about the protein locations. If the protein are not at the right time, the right place, it's not functional, and so on. So we will wait here, biologists, to find out all the ground truths and try to come up with a statistics model. It'll probably wait for 100 years and will not end here. So we try to do something. We try to see if we can help find out some gene regulation pattern without knowing too much, and that's why the statistical models becomes useful. Okay, so given that as a background, so now let's look at this diagram here. So we are considering the two genes. So for example, gene X could be the transcription factor I just talked about, and gene Y could be its target gene. And depending on the cellular state here, where the state here could be the state that the thyroid hormone is abundant or is absent. So then you will see the correlation pattern will change. And I'm adding a transition state there to indicate that the change could be in a smooth fashion. So if you can identify what state will controlling the pattern change, then you are done. But you are back in the question, because that's what if I ask biologists and they say, how would I know? And they will probably ask you, can you find it out from the data? And so we are coming back and forth. So I'm making a broad assumption here to make things going. So the assumption is that the state change is not just mathematical speculations or convenience, but it can be realized in certain way. That is, that there are probably some genes whose expression level change will correspond to the state change. In other words, when the thyroid hormone is high, there could be some genes acting up, and that will be reflected by the red bars. And when it's absent, this same gene probably going to be repressed, and that's here. So if you can find out certain genes whose expression are synchronized with your state change, then we are in a better shape. We simply look at the expression change of that genes, and then you can pop out the pattern changes here. Okay, so now there's a question here. If we would like to look all the possible genes and try to figure out, which genes are regulated in the way that I just described, then we have a huge computational problems I just mentioned. One way to go about the liquid estimate liquid association is to do the non-parametric regressions. Let me see. Oh, I have not. Sorry. Okay, so I'm sorry, I just jumped a little bit fast. So we cannot afford to use our eyeball to look at the 3 genes and try to see which one will work. That's not possible. So we need to have a statistics. So here is a little bit theory behind that. So let's first standardize the random variable. So each random variable represents the gene expression profile. So you have 3 genes, X, Y, Z, and they have mean 0 and variance 1. The correlation between X, Y is written as expectation of X times Y, of course. Now, since we are assessing the effect of the Z on the correlations, so let's first take the condition on the X here, and let's denote this conditional expectation to be Z of Z. So G of Z is an ideal summary about the association pattern between the X and Y when Z is fixed at the lowercase z. And because we are interested in the change of this association, so let's take the derivative, and that's the G prime. And finally, let's take the average, because you have so many Z's to look around. So this will be a first order approximation. And so I just defined it, okay? So it's not really the biology like it or not, but since I'm looking at this problem, so I just defined that. And so once you have a definition, then you have the data. So the question is how you are going to estimate. And for only non-parametric regression, that's not a big deal because this is only involve a derivative. So however, if you think a little bit harder, then you immediately have a problem because you have so many smoothing parameters to look. You know, one set of smoothing parameter can work for a certain set of the genes, but it would not for others. And you have the issue of whether we should set all this parameter at the same level or not. And so the issue becomes quite complicated. So eventually I give up that, although if you have a particular triplet, you can certainly do that. Now, I eventually use an idea I've done it, I use it in earlier my work that I was doing the relationship between the generalized cross validations and the Stein's unbiased risk estimate. And I'm pretty familiar with the Stein's lemma, and that can be used here. So Stein's lemma, because is a clever way of using the fundamental theorem of the calculus. And the way the fundamental calculus states is this: you have the function and take the derivative, integrate, and if x is uniform, then that's equal to the difference between h1 and h0. And as Charles Stein simply replace it by the normal. If you have a normal, then on the right-hand side, it becomes the x times h of x. So you can get rid of the derivative easily if you have normal distribution. Okay, so having that as a background, then we can estimate this quantity easily. It turns out liquid association equals to expectation of x times y times z. And this is how it goes. You first replace this g prime by gz, and then you uncondition it. There we go. So I apologize that quite different from many works present here. This is the only mess I have to say here. However, if you think hard about this, there are quite interesting properties that you can explore. There are, because it's a certain moment, so there's certain bounds, and about this liquid association you can figure out. And another important thing is that if well, this liquid association is a measure about the true nonlinearity of this 3 variables. If the x, y, z are jointly normal, then the liquid association score is going to be 0. Now, gene expression data hardly are normal. So what we do, well, we simply do a normal score transformation to force the z to follow the normal distribution. And I applied that to all the genes. And so this is the formula. So you have different conditions. You simply do this triplet product and take the average. And because the computation is so simple, so that it's still a heavy computing, but we can manage to do that for genome. For example, for the yeast, we can do it maybe in half a day and to run all the triplets. So having a measure about the change, the next thing is it really works. Does the result make any biological sense at all? So here I'm going to show you a couple examples. So the first example is the data from cell cycle experiment. So the biologists are able to correct data after synchronizing the cells. They spotted at different time points. So there's a set of 73 conditions on about 6,000 genes. So this is about the yeast. And I'm going to use that cell cycle data to study these metabolic pathways, which has nothing to do with the cell cycle. So that's interesting about the array data. You can always pop out some interesting things where the results are not on the data corrector's original expectations, because the cell cycle, usually the people use that to find the cell cycle regulators. And the urea cycle is complicated metabolic network here. I'm going to zoom in here. Well, I must say that the previous slide, when I do it many years ago, there's a misconnection. Even it was done from KEGG, but they simply make a careless mistake about assigning the genes. And I was lucky to find it out through some Googling and so on. So anyway, this is the urea cycle that is important for my analysis. The purpose of this cycle is try to maintain the proper level of arginine. And so when you think about the generating arginine, the raw material is glutamate and glutamine. So this gene is important. That's the ARG2. And there's also ARG1. And in fact, when I was looking at this data originally, I have no idea about this ARG things. It just pop out using some liquid association study, and I find out ARG1 and ARG2 there, and I was quite happy because it must be correlated. But it turns out there are much more to say here. So this is what I would like to show you why this liquid association does make sense. It's not just you are lucky to find the genes, but the way the correlation changes can be explained quite well. So if you look at this genes ARG2, and so glutamate has to be processed, and then eventually becomes this amino acid called the ornithine. And the process has to go this way here. Okay, it's not going this way. There's a direction here. So this raw material, this material, ornithine, have to be processed. However, there's a backdoor here, CR2. Oh, sorry. So that's the head. There's a backdoor. You got to shut it down because if this enzyme CAR2 is also activated, then the adenine will be degraded instantly, and so it would not much have to be processed. So you're in trouble. So if the cell want to synthesize arginine, then you would say when this one is up, this one has to be down. So you would expect a negative correlation between these two genes. Unfortunately, we do not see that from our data. So what we do is to use ARG2 as the X and the other one as the Y. And we simply play this liquid association game. We compute all the genes, 6,000 genes, and we simply rank them and try to find out if we can get any luck. And turns out that at the 8th place, with the most negative values, we find these genes, CPA2, which is right here. It's not the luck. Well, maybe it's 8 out of the 6,000. That's not bad. But as I said, liquid association is a measure of the change. So we would like to see if the pattern of change make biological sense or not. It's not just you find the high score genes. But so the score is negative, which means that the change is from the positive association to negative associations as this mediator genes goes from a low to high. Okay, you have the high and then goes low. And does that make sense? Well, the CAR2 and ARG2 are expected to have negative association, as we explained before, if the cell want to synthesize arginine. Okay, so CPA2 is high. Does that indicate the cell want to synthesize arginine? And the answer is probably yes, because in order for the arginine to go this way here, we need to have ARG3 to process it. However, ARG2 cannot function well without the other ingredient, which is the carbamoyl phosphate. And then carbamoyl phosphate would not be available unless you turn these genes up. Okay, so when this one is up, then the glutamine will be converted. It will be used to generate the carbamoyl phosphate, and then I will combine it with this one and to go this way. So that's how we think that when CAPA2 is high, is the indication about the cell's need for the arginine. Now for the other case, when the CPA2 is low, then they have a positive correlation. Now why is that? Well, the thing is that if the cell does not need it, which probably means that the arginine is already plentiful. Now, arginine is a charged amino acid. If you have too much of it, then you're in trouble. So usually the cell has a mechanism, which is to degrade it. So to degrade that, they activate CAR1. And also, they have to activate CAR2 also. So that's why under the situation that when this one is high, this one is also high. And because the cell does not need the arginine. So you have to have a way to get rid of arginine. So that's how you would expect the positive correlation. So in this case, the cell inside the body, they seems to be functioning on the way that this simple mathematical statistic is describing, and in the right directions. That seems to me quite amazing. And there are many other examples which I'm going to just simply skip. Okay, so in general, there are two ways of applying the liquid association. If you have a pair of the genes, then you can find high score liquid associations genes, and hopefully that this will be functionally associated with X and Y in terms of mediating their gene expression. Now, in many other situations, the biologist simply does not know what the gene's function is. So they will just give you the gene and want you to see, hey, tell me what genes may be related to this. So you can do with the association study to see which genes are correlated with that. But you can also do the liquid association study, try to see which pair of the genes has a high liquid association score. So we also rank that. Now, of course, the C part, the computation is more involved. So that's why we need to do a little bit of computing ahead of time and store it. So here's a few references that I've done using this technique. Now, I would like to show you how this things work amazingly in the cell line data. And the way, okay, so the data, well, NCI has developed a lot of the cell lines just to study how the cancer cells behave. You can apply some drug and see how many cells it killed, and in order to determine the effectiveness of the drug. So people has been using the microarray to determine the abundance genes in each cell lines and try to see some relationship between the gene expression and drug responsive. So we have that kind of the data available from internet, and I simply use it to illustrate some genes involved in the Alzheimer's disease. And I must confess that when I start with, I have no idea what to come up with. It just come out from some dinner conversation with my wife, and I was not focusing on anything. And he said, you must have Alzheimer's disease. And I say, okay, well, I'm going to look at what this gene is. So I look at the literature and find that this is the most important genes, is amyloid beta precursor. So I use that as the X and try to do some studies here. Okay, so people know that this Alzheimer's disease genes is, you find a little plaque, and the principal component, I mean, the major component is the amyloid beta peptide, the little things. And this little guys is derived from the this bigger protein through two enzymatic cut. One is called beta secretase, the other is called gamma secretase. And this is important, bear with me a little bit. So this is about the proteomics now. It's protein. So this is not a gene, but a protein, and this linearize it, okay, stretch out. And this part is the one get you the trouble. And from the bigger protein, you have to have two cuts, and each cut involving one enzyme. One is the, oops, sorry, how come it doesn't work? Shoot. One is the red, and one is the alpha, the other is the gamma, okay? Okay, so APP is causing the trouble, but it's also an abundant protein. Okay, everybody have it. So people began to wondering, what's the role of the APP? What was that? Its whole purpose is try to make you dumb when you're getting old or something. It's not possible. So this paper, Science 201, was the first to possibly identify its natural role. It's a physiological role. And by some sort of experiment, they lucky to find that the APP forms a complex with the Fe65, and this one is acetyltransferase tip 60. And remember that I mentioned the histone transferase in the beginning. So they deduct that this gene's role probably is involved in the transcription regulation. Okay. All right. Now, so because APP has a protein interaction with these two genes, as this one suggests, so I'm going to study how their expression look using the 60 cell line data. So unfortunately, there's no correlation. The correlation between FE65 and APP is essentially 0. And again, this one and the APP is also 0. So that's a perfect example for trying my method. So I take one, this APP is encoded this guy and so on. So liquid association, if you look at the high scores, normally my computer will pop out the top 20s. So in this set of the genes has about near 9,000 genes. So 20 out of 9,000, I've been able to find this BAC2, which is a beta site APP cleavage enzyme. So I'm quite happy with that. But you may ask, what about the others? Well, so I have this, and this one is quoting the others. And the result is even better. This enzyme called the PSEN is on the second place and is encoding presenilin as a major component of the gamma secretase. So, the data really helped me, and that's helped me to publish the papers. And I present this story to a colleague of mine who was studying the multiple sclerosis, and actually a postdoc first. And he's so excited, he grabbed his advisor or supervisor with me, and he's a genuine physician. So they just sit with me, and we go through this Alzheimer's disease case, and he says, now let's try your method on multiple sclerosis. And we have on our data set a set of the expression. So first we try on the 60 cell lines, but we need to have two genes. Okay, sorry. We need to have the two genes. So one gene, let me see, what is one gene? Okay, one gene is myelin-based protein. That's the gene which is producing, which is abundant on the myelin. Myelin is a protein wrapping around the axons. And at that time, they were interested in this gene called the PRKCA. They found it from some genetic mappings. So he is trying to say that how your method helped me to find more genes based on this. So we use one as X, this one as Y. And using the data, well, this is not the cell lines data, because 60 cell line data at that time, we do not have these genes. So we use another data set, which is similar in nature, but it's a data set collected from different people's different bodies, brain, liver, and so on. And it has about 150 conditions. It's quite huge data. Okay, so we again play that, and we find that these genes called SLC1A3 pop out at I think the first place or second place, and the physician's eyes sort of shines because he recognizes these genes. He says that, well, this one sounds familiar, and it turns out this gene is belonging to one region on chromosome 5, right? But they have not studied these genes before because it's right on the edge of a big intervals that the previous gene mapping method been able to locate. the segment is so big. So when they do the fine mapping, they simply take a little chunk of it and analyze it. So at that time, they analyze on the, say, right end, but this one is sitting on left end. There's a big gap in it. And the postdoc was taking 75 markers from that regions. And because we find these genes, so they say, okay, why don't we give it a try on this one? So they take another 5 markers. This is all SNP markers. So in total, there are 80 markers, and they have family data using about 300 patients from the database. So the result out of these 80 markers, they find only 2 markers that are significant, and one of them is sitting right next to, adjacent to this LC1, S3. So it is very happy. How about the other? Well, the other is this gene here, CTA2. So on this chart, this is the chart that we use by using the liquid association, both the liquid association, which is used in this triangle here, and also the regular correlation. So using the correlation and the liquid association, we find that these genes are related to this. And but we have a big trouble publishing the data because the confirmation, the data size is too small by comparisons. 300. So we try to sell it as a methodology papers, and eventually we go down to genome biology, which is not bad. But still, they have a kind words on us, but they still want us to do more data, confirmations. And my collaborators, for some reason, hesitated. Those genetics, once they have certain findings, if you collect more data, you either reconfirm it, or the whole thing will be a negative finding. So you stretch out things. So usually they try to publish it, no matter what. However, fortunately, when the referee report came back, which was Salt Lake City, which was last year, I was sitting in a hotel room, and I did not go to the meeting at the time because I have the jet lag. And there's a newspaper put outside of the door, and the headline is multiple sclerosis gene find. Multiple sclerosis is popular in the white Caucasians, and so that's why probably got their attention. So I was curious what genes they do find, and they refer to these genes, not my genes. Otherwise, I would be super famous right now. But they find these genes are IL7R here, and in fact, on the last year, there are 3 all the standing papers are about this, and 2 are in Nature Genetics. And the one they actually point out is this paper on New England Journal of Medicine. It involves huge studies. You can see how many? They have 6 of 9 families, 2, 3, 22 case, 7, 8 control, and they're all done on this FE chip. So it's a very heavy investment, just burning the monies. And how many genes they find? 2. One of them is the IL7R. Now, this gene sounds familiar to me, although I've done this a long time ago. If you look hard on this, where is IL7R? Got to be somewhere. Right here. Yeah. So it's derived from MBP and SLC1S3 using this data, NGF, which is the data I described before. And it's one of this long list we found here. So that seems to be a success story. And then we look at a little bit closer to our data. And in fact, the marker that we use is not contained in the FE SNP. However, we use their data. And this is how we got the paper published. Because we go back and look at the data provided by this New England Journal, and we find that there's a score. We compare the SNP near IL7R in that region, and we find that there's a marker near our gene region which have the score even better than IL7R. And in fact, IL7R and our marker and our genes are very close. They are only 1 megabyte. Did I say that? Yeah. It's within 1 megabyte region, so that's short distance there. Within that region, our score is even better than that one. So you may wonder why they use IL7R instead of the other. So there's some story behind it, because the IL7R was some group some investigator among this big consortium are already interested in that genes. So that's why they give a higher priority, and they confirm it. But in fact, that the reason that I'm so impressed about IL7R is that when I dig out my emails, I find this one sent to my colleagues. And I say that this one is interesting, and they never pay attention to my email. And this email was sent much earlier. Did I say right? So anyway, so I think my time is about up. So I'm going to stop here. I have some other things. There are some minor statistics things. If you are interested, then I'll talk more about it. Otherwise,
questions?
yes.
I have a very basic question. You were looking at the expected value of G prime of Z.
G prime of Z, right.
Right. It seemed to me the natural thing to look at is how the conditional covariance between X and Y varies with Z. That isn't the conditional covariance of X and Y as a function of Z, because you'd have to subtract off the expected value of X given Z times the expected value of Y given Z, which are both very hard to estimate. And you could have the conditional covariance be exactly 0, and yet the expected value of G prime of Z is not 0, because you don't subtract off the means.
Right.
So I'm very confused what your statistic is and why it should work.
Well, why this works? Because nature works that way. So that means that.
Why does nature work that way if it doesn't have to do with the conditional?
Well, that's somebody was saying that the two views. One is the words present as it is. The one is that. But let me explain to you that what's the confusing things is this. What we are looking at, we have to think about what we are aiming at instead of what it mathematically should be. So it's the language problems. So if we look at the how people use the correlation to reflect, and that has to do with the question that Peter was asking before, why this correlation is useful in explaining this similarity. So, in fact, there are more than one measure that people use in that, but this is just the correlation. If you standardize everything, then the mean is 0, and this mean is 0. So a positive correlation looks like
the marginal mean.
Right, this is the marginal thing. So means that marginal, well, what's marginal means? It's just the different conditions that you combine together, right? That's so each dot represents all the possible condition you have here, and the mean usually the biologists we will tend to interpret as the background values or the standard values. So that means that if you average out all the possible situations, that's where the gene's expression should be. Okay, so as a ground value. So genes higher than that, that's upregulated, and this one is downregulated, and so on. Okay, so if you have the two genes, they are both up, then you have and both down simultaneously. So that's how the biologists tend to like this idea. Now you can have another measurement starting from somewhere here. So the way the origin, the choose of the origin need not necessarily to be here. You can start with certain places and define your up or low regulation. So if I'm willing to set up a value and consider that which is up and which down, so this part is fixed. Now, among these conditions there are some corresponding to the larger value of z. So I'm going to highlight it here. Maybe it's these conditions. Okay. So now, when you look at these conditions, as a biologist, are you going to say it's up or co-regulated or not? You should not, because you already fixed this one as your sort of the ground levels there. When you look at the z, if you still think of the mean as the new means as the value to compare with, then that's violate the biologist intuition, because you have many ways of talking about when it's up or lower regulated. So that's why it's not only mathematically, it makes things simple, by dropping this EX, but also makes the biological sense, because we are using the fixed values here to represent whether a situation is considered as the up or low regulated, and it was whether synchronized or unsynchronized, which is like this pattern. Okay. Yeah. Yeah,
It's a lot of words, but I don't get why one would have chosen that statistic.
Well,
Except that it's
so
famous, except someone forgot to read your email.
well, you can well, you can ask why you need have to use the correlation change to model a biological phenomena. That's the fundamental questions. Right? You can think that mathematically one should do that, but there are many ways to describe the world. Now, why do you need to have to use? I know what you are talking about. You need to have this conditional z, and you want to take the derivative, right? So what does this guys do? Now, that's the question. I don't know how to explain this, so therefore I choose this simple one here, because in order to do this, you have to shift your new means here. Okay, and shifting the new mean, I'm not quite sure how would that.
Well, it's just conditional covariance given z in a world where we don't have to worry about other confounding factors. If z, for example, is if neither x and y cause each other, but z is a common cause of the two, then when you condition on it, you'll get the covariance. In other words, you can tell causal stories, true or not, under which covariance of x and y given z would be 0 or not 0 and so forth. It's just, I don't quite understand the story about the underlying data generating mechanism, the causal graph under it, under which your statistic, I couldn't predict, knowing the data generating mechanism, the topology of what causes what, whether your statistic was going to be positive, negative, or zero. That's, well, I could about something like, does the conditional covariance.
Well, I can turn the question to you. I can say that I simply do not understand the mechanisms that why the covariance change would make sense. I mean, give me any example that why it's a reasonable causal effect because
Well,
you are
so-
simply
so-
you are simply throw away the mean effect, right? The difference between this expectation of xy given z, you take the derivative, and the covariance of z, which is e of xy minus this. So we are starting the change of this quantity, and you say that this change of this quantity will be more meaningful. I don't know why this one would be more meaningful. I'm not saying this one is not meaningful, okay? But you are telling me that this one is more meaningful. That's why I don't get it. Yeah. I think there will be certain situations. There's certain situations that this could be meaningful. I'm not saying that you cannot speculate it. But what I'm explaining here seems to be quite natural, because that's the way that you fix your baseline and then try to determine when it's up or low regulated. And that's how it, yeah. But I cannot incorporate the change, simultaneous change of the shape and the level. But there could be some situations better to separate it. I'm no doubt that could be the case. Yeah.
A little bit. Does anybody else have any more questions? I have a little question, but I know you're all desperate to go. But so I guess another way to think about this might be to use, say, maybe a latent variable model, something like, for example, mixture factor analysis might be another way to go, which would try to find actual correlation structure there, which otherwise, perhaps on the marginal correlation structure is 0. Do people look at those kind of techniques, or?
Maybe, but we have a paper, I probably not listed, that we actually using what you do, try to model the transcription factor, the expression of the transcription factor, and there possibly several targets. And then we will try to associate this simultaneously. So we use a mixture model, so there will be 2 states. And then we put out one state, you have certain regulation, and so the slope will be changed between on that. And the problem with that is that you can try to dichotomous the state, but that kind of the things does not allow the smooth transitions, right? So you can have more complicated models, but the computation becomes hard. So we have little papers on that, and it seems to be working fine. I mean, there's no problem on first order approximation using the dichotomous one. The computationally, this one is much simpler than this one. Yeah. So, but if you have a specific network to do it, and that's probably a good way to do it too, and it can accommodate the simultaneous change. So I'm not fully against the modeling. Now you want to think about what things can generate it. That would be fine. Now there's another advantage about this method, that is, since we do the normal score transformations, so it is robust to the outlier, which is kind of important, because once you screen all this 6,000 genes of that, quite often you've got to pop out an outlier. We find those correlations usually close to 1, and in the beginning we're very happy, but it end up with it's all dominated by one big numbers there. This is kind of typical. So the robustness has come out just by accident.
Okay, any more? Or let's thank the speaker once again then.
Thank you. We have a harder time on fighting that if a referee are questioning.
Did the referee say it?